Thank you. Thanks for having me. So today I would like to talk a bit about the Michigan Imputation Server and what we did lately with that and Nextflow. So a very short introduction to the server. So as you might know, the Michigan Imputation Server is a free service for doing genotype imputation, and you see that on the top right corner in this picture. There are two lines. The first line is a fully sequenced sample, or a fully sequenced sample in green, and in the second line you see microarray data in red, and you see these gaps in between. And basically what genotype imputation is doing, it's filling these gaps for us. There is software out to do that, and as you might also know, that's really a key component for all the GWAS around the world, and many are using our server, and you see this in this graphic where there's the researcher, and the researcher can upload the microarray data to our server, and on our server we have different reference panels available. These green lines, many of these green lines, so they are normally not publicly available, and we are doing the imputation and sending the data back. Okay, we published that together with our colleagues from the University of Michigan and the Zurich Institute in Italy already 8 years ago. But I can tell you the real work started after we published this paper. So basically, from an end-user perspective, imputation is quite easy. The only thing what you need is your microarray data in BZF format and a reference panel, if this is publicly available, like the 1,000 genomes reference panel. Then you can do the imputation locally and you get your results. Minimac4 is the system coming from the University of Michigan. Of course, this can take days or even weeks, depending on the size of the input data or the reference panel. So when we go a step further, then when thinking about the full imputation workflow, it gets a bit more complicated. You not only need the imputation itself, you also need a phasing step, maybe also a quality control step, and if you parallelize that, at the end you need also a compression or encryption step. When it gets really complicated is when you try to provide a full imputation workflow as a service to the community. And then you really have to think about many different things, like provide different reference panels, provide different workflows. On the server, we have HLA imputation. We provide also polygenic risk scores, so different applications. We have to think about web service security. Of course, we have to think about testing and parallelization. And basically, this is what we did in 2015. And you see in our stats that we were quite successful with this approach. Right now, we have around 117 million imputed genomes. We have a large user base and normally also a lot of running jobs. And it's really exciting to see that also the TopMat imputation server, which runs by people from the University of Michigan, also uses the same architecture as we do, with, as you can see also here, over 60 million imputed genomes, human genomes. Okay, let's go back to the architecture in 2015, and the workflow is based on a technology which is called MapReduce, famously published by Google in 2004, and we used that technology. Basically, we use the Apache Hadoop framework, which is an open source implementation of that, which consists of two parts. It's the MapReduce framework and the distributed file system. And as you can see in this picture here, basically our server consists of two MapReduce jobs. The first one is for quality control, the second one is for phasing and imputation. So we split our file in 64 megabyte blocks. Then each of these blocks is going to a map task. We're calculating statistics, and then we are reducing that per chromosome. The second one is a bit different. It's a map-only step. Basically, what we are doing here, each map task gets exactly one line, and then which includes the configuration of one chunk. We're doing the phasing, imputation, and then the merging afterwards. Yes, the yellow elephant is the thing from Apache Hadoop. And this is how we started in 2013. It started under my desk with 3 nodes at this point, and Hansi, my colleague, shows you the architecture from back then. What I really have to say is that this approach worked out really, really well. So we really have almost no unexpected job failures. there are really strong components included in the MapReduce framework, like the distributed file system. We were quite resistant against node failures, a nice caching mechanism, all good. We had a throughput of around 1 to 2 million genomes per month, and as you can imagine, many challenges along the way, but yes, it was really successful. But when everything is great, why we should move away from MapReduce? And the answer to that is probably there are several things which I could talk about, but the key issue was really not Hadoop itself. But over the years, our cluster grew. We have, as I said, a lot of imputed genomes. So we had 40 plus nodes. We still have 40 plus nodes with different number of CPUs and memory. And what we needed to manage that is a good cluster management tool. For example, when adding nodes, when changing the configuration, when doing updates of the underlying MapReduce framework. And for that, we used Cloudera Manager. And that worked really, really well. But what happened to us is that they changed the license model, and this was really a problem for us. Because we are an open source project, it's a free service to the community. So we had a deadline in our head, and this was the upgrade of the operating system. And we knew until this point we really need a solution to that. Okay, so after many, many thoughts, after many, many checklists, we reached this point of no return. And you see here, Lucas took a picture of that. After 10 years successfully running Imputation Server with Hadoop MapReduce, we shut that down. But I can tell you that really glad to let you know that we fully migrated the complete Michigan Imputation Server workflow, including validation, QC, phasing imputation, compression and encryption to Nextflow. So now the benefits come with Nextflow. We now can support full local and cloud deployments. We even have Nextflow pipelines available to create local reference panels, and we bring it one step closer to a community project. All the source code is there. I'm not talking about the pipeline at all, but the source code is available under this URL. Okay, so a big thing, what we had to think about was testing. So all our tests are based on NFTest. We migrated all our JUnit tests from Java to NFTest. We even created a new NFTest plugin, which is called NFT VCF. So that helped a lot when migrating the complete source code to Nextflow. We also did a lot of stress tests. So we were submitting a lot of jobs with a lot of genomes via the API. You see this in the top, in the right corner, in the bottom right corner. And the other picture, what we also did is we simulated arrays from whole genome sequencing data to determine really the imputation quality in this XY plot, where you see the minor allele frequency on the x-axis and the imputation R-square on the y-axis. So that was really crucial for our project. So just 2 or 3 screenshots on the Michigan server. You see here, it's a graphical user interface. You can log in. You can then upload your VCF data. You can select the reference panel. There are also some more options what you can do in the interface itself. But then the job gets queued on our side, and at one point it gets running. You get feedback. And if the job has finished, you get an email notification from the system, and you can basically download the data, and after, I think, 5 or 6 days, the data is completely deleted from our server. So we are sending a password via email, and then users can download the data from here. Okay, so what I talked until now is that basically we migrated the Michigan server to Nextflow. But as you can see, we have a graphical user interface here. So the question is, how can we do that? And for that, we developed a system. Lucas, you know, Lucas from yesterday from NFTest. So basically what CloudGen is, so it's a system called CloudGen, and basically what it is, it turns a Nextflow pipeline into a graphical user interface, into a graphical web service. So basically you can connect your pipeline with CloudGen, you can monitor your jobs, you can it renders tasks, and it's all on cloudchain.io. Probably I cannot talk too much about that, especially when seeing the clock. But the fun fact here is that when we firstly presented that was in 2015 at the massively parallel sequencing workshop in Uppsala, and that was also the first time we learned about Nextflow. So it goes already back a long time. So this is CloudChain, and basically what you can really do with CloudChain, you can connect it with your Nextflow pipeline. So I talked mainly about the Michigan server, but you can also use other Nextflow pipelines and connect that. Even what we did, I think it was last week, you can even combine Nextflow pipelines and present that as a service to the community or as a private service to your company or your institute. So what we, for example, did, we combined the Fetch NGS pipeline with the text profiler by writing a YAML configuration. This connects the pipeline to CloudChain, and then you have a graphical web interface, and you benefit from all the advantages, from all the things we learned over the last 8 to 10 years with the Michigan server. So I just have, I think, 2 or 3 screenshots left. So what we did basically is this is this great pipeline. It's not coming from us. It's also about imputation, more like coming data from low coverage sequencing data. And we checked out this pipeline, and we thought that would be a nice idea to integrate that as a service. So what we did is a proof of concept. You see on the left, on the left picture here, the YAML configuration where you can combine a pipeline to CloudChain. Then we have a rendered graphical web interface. You can, again, select the data. You can upload the data. You can select the reference panel. And then the pipeline runs. By using the plugin from Nextflow, we can monitor the jobs. We can also render output. Then the job has finished. You see here the output of the phase impute pipeline. And now when I click on the ModiQC report, for example, then I can have a look at that. Just a short and quick demo of that. So what we did in our work is we successfully migrated the Michigan server to Nextflow. We are really happy, and we hope that many of you benefit from that. And I also want to say that we put the CloudChain preprint 2 days ago on bioRxiv. So if you like CloudChain, please check that out, and please get in contact with us if you like it. And acknowledgements to the complete team, especially Lucas from Innsbruck. We're working together for many, many years, and we hope we can continue that. So thank you.